Hi everyone, it's Dr. B here, and this is Physical Principles of Quantum Biology, Chapter 7, Enzyme Catalysis, Quantum Fundamentals. I'm Nathan Babcock, and this is from our book, Physical Principles of Quantum Biology, with my wife, Brandy Nicole Babcock. And today we will be finishing up part 1 of the book, Chapter 7, on the quantum fundamentals of enzyme catalysis. So, let's continue. First of all, what is an enzyme? Enzymes are specialized biological catalysts, primarily made of protein, that accelerate life-sustaining chemical reactions by lowering the energy required for them to occur. However, enzymes do not complete these chemical transformations alone. They have cofactors. What is a cofactor? Well, enzymes recruit molecular cofactors to act as tools that carry out specific tasks like moving electrons or modifying functional groups, chemical changes. And cofactors may be either metal atoms or organic coenzymes like NAD or FAD. So if you look at this enzyme, monoamine oxidase B, and we take a look inside, we can see its cofactor, flavin adenine dinucleotide, otherwise known as FAD. Now, why do we care? These molecular partnerships are essential to ensure that processes like energy transduction and DNA repair happen in a timely fashion, in, say, microseconds, rather than spontaneously on their own, which could take centuries or even millennia. How do they do this? Well, there is a nice semiclassical picture of how enzymes work. First of all, the idea is that the rate acceleration, that is how the chemical reaction gets sped up, is primarily accomplished by lowering the activation free energy via what's called electrostatic preorganization. So if we look at our typical reaction chart, it has a reaction coordinate and an energy axis that shows how the reactant substrate, the reactant molecules, are transformed to the product molecules, but it has to go over something called a transition state first. And the enzyme works by lowering the transition state energy, making the reaction, how do we put it, easier to occur. So this electrostatic preorganization transforms the energy of the transition state, and this is set by the protein fold and the catalytic architecture of the active site, the cofactor. Now, this semi-classical picture accounts for much of our knowledge of enzymes, but in many cases, there are also quantum contributions that play a role, such as vibronic coupling between nuclear and electronic coordinates, non-adiabatic dynamics, tunneling, in which case it goes through rather than over the barrier, delocalization, resonance, and spin effects. And in fact, complex catalytic cycles can both involve adiabatic, by which we mean essentially static barriers, quasi-static to a physicist, and non-adiabatic regimes, which involve ultrafast dynamics, such as photochemical or electron transfer events, like we see in the DNA repair enzyme photolyase, which we considered in our last lecture. Now, if you look at another enzymatic energy chart, here we have a reaction coordinate and an energy axis. And if this is describing, for example, an electron transfer reaction, we're going to have an electron going from the reactant surface to the product surface, traveling from an electron donor to the electron acceptor, say. Now, what's interesting is, even in our semi-classical picture, there are underlying static quantum effects that are innate to the electronic structure of the biomolecule itself. And even the static, seemingly classical structure of the molecule, the protein, the enzyme, involves typically delocalization, sometimes tunneling, and even resonance stabilization effects that aid the electrostatic preorganization. And so even when we account for the underlying quantum structure of matter, we find that this so-called transition state stabilization picture of the enzyme reaction is still not complete for a perfect understanding of enzyme catalysis. And so if we look at something like an electron transfer reaction, the rate, KET, is given by what's known as Fermi's golden rule. We have a constant 2 pi over h bar times a matrix coupling element, the transfer element that couples the donor to the acceptor, squared, and what's known as a Frank-Conon factor. And the dynamics, the true electron dynamics, are accounted for by that TDA, the electron transfer factor, the coupling matrix element. Enzymes and photoreceptors often employ quantum dynamics in physiological processes such as vision, photosynthesis, and even certain ground state organic reactions where dynamics can play a role. And dynamics have been properly defined in the quantum mechanical sense by Warshall and co-workers previously. So in order to understand enzymes, we have to think of both the static and the dynamic contributions is what is ultimately important here. So if we want to consider enzymes as quantum biological catalysts, first of all, enzymes can increase reaction rates by factors that are enormous, 10 to the 6 to 10 to the 12, or in extreme cases, even as much as 10 to the power of 26. That's a lot of zeros. So the most informative examples of the quantum effects are enzymes in which multiple quantum effects interact. So first of all, we have the underlying quantum physics of the electrostatic preorganization itself. The protein scaffold pre-aligns dipoles, quadrupoles, octupoles, the whole dielectric structure of the protein to stabilize transition states, lowering classical chemical activation barriers through favorable electric field orientations. Now we have the non-adiabatic dynamics, the true quantum dynamics that involve tunneling, photoexcitation, and possibly spin-coupled electron transfer effects that dominate the ultrafast aspects of the reaction and operate beyond what is called the Born-Oppenheimer approximation that treats our system as essentially static. And finally, our third key concept is basically the structure of the enzyme systems themselves. There's a variety of different structures that we see, these recurrent motifs in quantum biology, metalloporphyrins, flavin-dependent enzymes, and other structures that are frequently seen, for example, in electron transfer and electron bifurcating complexes, just to name a few. So, to start with the fundamental concept of the electrostatic preorganization, we can think of classical electrostatics. It's just a charge distribution, positive and negative. But at the molecular scale, we also need to account for true quantum mechanical effects, such as delocalization and resonance. Now, what is delocalization of an electron? For example, it is the electron's ability, so to speak, to be in a superposition of different places at the same time, a spatial superposition. The spread of the electronic density across multiple atoms, molecular regions, bonds, or structures in general creates long-range interactions and enables the functionality of the charge distribution in many important cases. Additionally, we have an effect that can play a role called resonance stabilization. And this is a quantum effect that enhances the stability of a molecule by lowering the molecular energy, and it's achieved by the superposition of multiple valence electronic structures. Now, the concept of delocalization and resonance are not identical, as electronic delocalization can stabilize or destabilize depending on the context. And both of these concepts are crucial to understand reactivity, spectra, aromaticity, of course, charge resonance, and other long-range coupling effects in chemistry and biochemistry. Now, before I get into the quantum dynamics, I want to take a moment to consider the classical dynamics, stochastic, structural dynamics, and conformational gating. These are classical movements of the protein that may control quantum effects. If the protein is moving, wiggling and jiggling, or opening and closing, the Change in the structure, the distances involved can influence electronic couplings, as for example, a donor and acceptor molecule get closer together or further apart. So this can also regulate, in addition to electronic states, protonation states along reaction pathways, the movement of protons, and potentially even synchronize structural motion in changes in the electronic correlation. And so the question of the degree to which these structural dynamics don't just gate chemical reactions, basically turn them on and off, but modulate them in a truly dynamical way remains a topic of open debate. But going beyond the structural effects of the shape of the protein itself, we also now have what are called these true quantum dynamic effects, non-adiabatic effects, and ultrafast photochemical effects. Now, these are directly related to, for example, electron transfer couplings or excited state couplings. They can include vibronic, excitonic, and polaritonic couplings, and they enable transitions at what the chemists call conical intersections and avoided crossings, where these true ultra-fast quantum dynamics occur. Now, as we move on to the picture of the cofactors themselves, Bertin and Balaban has very nicely said that Darwin at the molecular scale cannot escape the rich and grainy atomic structure of matter. And so if we consider the way life has been shaped and evolved over the millennia, we see that a number of structural motifs have appeared. For example, the indole ring of tryptophan. If we combine a couple of tryptophan, we get, for example, a tryptophan-tryptophyllquinone, flavin mononucleotide, where now the double ring structure has been replaced with a triple ring structure, a tricyclic, if you will. And as I mentioned, even metal-containing structures like the plastocyanin center here, where a metal atom is coordinated by aromatic residues, and this controls its catalytic properties. And you can look at the paper I've cited here to see an extensive list of various biological cofactors that are now known today. But what's interesting is the relationship between their chemical structure, their electronic structure, and the functional properties that they enable in biochemistry. So if we look at, for example, a common quantum effect, electron tunneling in heme dimers. If you have heard of hemoglobin, the oxygen-carrying enzyme in your blood, you are familiar with the idea of the heme group. But hemes play a lot of different roles in biology. It's a porphyrin ring with an iron atom at the center. In this case, we see an electron being transferred from one heme to the other. Now, what's interesting is in the enzyme cytochrome B03, it has a diheme copper active site in which a pair of heme groups are coordinated with a nearby copper atom as well. So we have three metal centers. Now, there's a physiologically relevant tunneling mechanism whereby quantum mechanical tunneling controls the physiological electron transfer step during respiration in the quinol oxidase enzyme, cytochrome B03. Now, studies have shown that coarse-grained methods that average over the atomic and molecular structure of the intervening matter are not adequate to describe the tunneling effect, and thus methods that consider simply that average packing density of the medium can underestimate key electron transfer parameters. So the insight here is that a true atomistic approach is required to fully understand the mechanism of short-range electron transfer processes like this one. If we consider another metalloporphyrin, chlorophyll, which enables photosynthesis, there is something called the photosynthetic special pair. This is not not what's usually considered the antenna that absorbs the photons of light, but it is the reaction center where the light-induced excitation produces a charge-separated state that goes on to produce truly the biological electricity that powers all life as we know it. So there is a quantum optimization at the special pair where strongly coupled chlorophyll dimer, as you can see I've labeled here, form a delocalized excitonic and charge transfer state prior to charge separation. And now this condition is optimized in order to ensure that the charge separation event occurs. And when the electron separates from its so-called hole, the positive counterpart to the electron, that it travels in the right direction. And research has shown that charge resonance interactions, vibronic coherences, and other important quantum effects, in fact related to what's known as a phenotype quantum resonance, are likely to be important to controlling the charge separation event in these systems. And so something even as well known as the photosynthetic reaction center has a rich mechanistic complexity, and quantum simulations are needed in order to reveal the interplay between delocalized excited states, the vibronic coupling, and the protein environment itself that often plays an important role in controlling systems like this. For example, during photosynthesis, the overall light levels will control the structure of the entire antenna, which must adapt to gather more light in low light levels and actually to release excitations to quench them through carotenoid molecules, for example, in high light conditions, which would otherwise be overwhelming to the reaction center. So there's a really dynamic responsivity of these kind of systems to their environment in order to control how much energy, in this case, is going to the reaction center versus being quenched to the surrounding environment to prevent damage. Now, there's another interesting enzyme I've studied in my research on quantum biology, MAO-G, a long-range charge resonance effect is carried out in this enzyme during its catalytic process. This enzyme is responsible for catalyzing actually the creation of active sites of other enzyme. It has a cofactor that's used to build other cofactors. And so this is another diheme enzyme. It has a pair of hemes that stabilize a high valency iron 4 state. So this is proposed to use a tryptophan bridge delocalized radical. It's supposed to be a type 3 charge resonance, although this is still under investigation. And, with all the jargon aside, the basic idea is that an electron is delocalized across the two heme sites. And this delocalization and the long-range electron transfer that it enables in coordination with structured water and various amino acid side chains enables a proton-coupled electron transfer event that actually allows it to perform catalysis at a distance. And so the actual active site doesn't even need to come in contact with the chemical process that it's catalyzing. And the distance enables it to do this more safely and efficiently because the redox potential of this system is so high. Remember, it is catalyzing the chemical steps that are needed to create another cofactor. And so if you're building a system that carries out chemical reactions, it's like basically, I don't know how to put it, has to be supercharged. The idea is that the system is building another system that is also going to be chemically active. And so it actually has to have an even higher oxidizing reduction potential in order to properly carry this out, which carries higher risks, the potential of damage even to the enzyme itself. And so it's really interesting to understand how the protein structure controls the various quantum effects, quantum delocalization, and other spin-related effects in this kind of system. And so this presents a general principle of metalloporphyrins, and in fact, as we'll see, many enzyme cofactors in general, that the metalloporphyrin enzymes use the porphyrin macrocycle as a tunable electronic platform with delocalization properties that are matched to the chemistry that they catalyze. So whether you just want to transfer an electron, whether you want to create a delocalized state, or even a strongly oxidizing or reducing state, the protein environment actually controls the electron structure of these often repeated looking, similar looking cofactors in order to get different kind of catalytic effects out of them. So I'm also going to suggest that in addition to these metal centers, that organic cofactors such as flavoproteins may be thought of as quantum biological prototypes. So for example, the well-known protein cryptochrome contains a flavin cofactor, FAD. So cryptochrome and the related protein photolase are involved in light reception and DNA repair, as well as electron transfer and in fact, flavin's involved in something known as electron bifurcation, which I'll discuss at length in this talk, in order to produce a variety of different forms of catalysis. Why are flavin so ideal? Well, they're highly tunable, they're air tolerant, they have spectroscopic properties from an experimental point of view that can be used to probe the rich quantum phenomena that they are ultimately enable: tunneling, delocalization, spin dynamics. So they're both seen frequently in biology and physiology, and they're handy to study experimentally. So they're really prototypical, both in terms of the physiological function that they enable and in terms of our ability to study them in a lab. Now, they're not that easy to understand, though, because despite the relatively simple structure of the flavin, it's extremely sensitive to its environment, and it has functional properties that are still difficult to describe quantitatively, even after decades of research. In fact, even the consensus on the best simulation methods, quantum simulation methods, is not fully established. So if we consider flavins, for example, the FAD, flavin adenine dinucleotide, in depth, it has this tricyclic isoalloxazine ring. This is what gives it its flavin character. Ribitol and ribose sugars that now connect to an adenine structure, and of course all linked by a pyrophosphate. So this is just one type of flavin, one of my favorites, but all flavins, including riboflavin, vitamin B2, flavin mononucleotide, and of course FAD, feature that isoalloxazine ring with correlated and delocalized electrons. The overall structure give it a kind of functional versatility. It's been called life's most versatile molecule because it's sensitive to light, magnetic fields, electrostatics, oxygen, pH, surrounding protein dynamics, and in even local chemical modifications. It plays a wealth of physiological roles, as I've suggested, from respiration to bioluminescence, light reception, circadian regulation, DNA repair, redox sensing, and other areas of physiological regulation. So it really has it's a workhorse with enormous range of applicability, and it has interesting spin chemistry. Photoactivated FAD forms a pair of spin-correlated radicals, enabling even magnetically sensitive chemistry. But despite its wealth of applications, the understanding of flavins, like flavin mononucleotide, is still somewhat limited. For example, the electrostatic preorganization, hydrogen bonding, solvation, and protein dynamics can all influence its spectra, redox potentials, and reactivity, in highly context-dependent ways. So it's not always easy to predict in advance what it's going to do. Of course, this gives it its functional versatility and makes it interesting but challenging to study. There are open problems in terms of how it deals with oxygen, how it interacts with oxygen. Although it has unparalleled versatility in oxygenation chemistry, flavin oxidation by oxygen, O2, in fact, remains poorly understood, and there are no general structural rules to predict how oxygen reacts with diverse flavoproteins. In terms of photophysical modulation, its interaction with light, natural and synthetic covalent modifications, chemical changes, can adjust flavin reactivity, simply by modifying the structure itself. And the choice of a quantum mechanical approximation in order to study those changes can critically affect photophysical reactions. Well, as a physicist, of course, I would like to think that if I have a good approximation, or a set of approximations, that they're all going to make the same predictions. But the fact that our choice of the approximation affects our predictions really means that the approximation itself is in question, and we may need to resort to a more fundamental approach, perhaps that, as much as possible, abandons approximations entirely, back to first principles. And so predictive modeling and methodological benchmarking are still needed. What's interesting is failures of sequence and structure-based methods, our inability to predict the activity of the molecule by its structure alone, reveal that electron correlation-based methods are needed to capture not just the simple semiclassical structure of the molecule, but fundamental aspects of the electronic structure, delocalization, and environmental coupling are going to be essential for actually predicting flavin function in physiologically relevant conditions. And so, for anyone who thinks, quantum effects, are they trivial? We see that they're highly non-trivial. They're actually at the forefront of research in quantum chemistry today, and they still challenge us to understand exactly how to model, even a relatively small organic system like flavin. So if we now scale up to a flavin-containing protein like the electron transfer flavor protein. Here is a canonical ETF here. This is human electron transfer flavor protein. It has an FAD cofactor and an AMP cofactor. These are stored in two different subunits of the protein. And in this case, the purpose of the protein is to transfer electrons from fatty or amino acid oxidation steps to the respiratory chain, where metabolic electron transfer occurs, where the energy of life is transformed. Now, in addition to the canonical electron transfer flavor protein, which, as the name suggests, just transfers electrons, there's something called a bifurcating electron transfer flavor protein. Now, in a bifurcating system, it doesn't have the AMP, it has two FADs, and this is correlated with the overall conserved structural motif of these enzymes. And the pair of FADs enable electron bifurcation chemistry, which I'll describe in a moment. The idea is that during an electron bifurcation reaction, for example, NADPH might reduce FAD to FADH minus. The flavin has now been given a pair of electrons and a hydrogen nucleus. Now, one electron is going to be sent from the FAD uphill, for example, to a ferredoxin, while the other is sent electrochemically downhill, for example, to quinone. And this is important because it allows energy transduction. Basically, you get two electrons with, we should call it a moderate amount of energy. And by sending one uphill and the other one downhill, one electron gets a lot of energy, while the other electron gets less. So they come in sharing the energy, but they go out of the enzyme in a symmetry breaking effect that allows one electron to take much more of the energy than another. Anytime you talk to a physicist, they'll tell you these kind of symmetry breaking effects are the most interesting areas of physics. And in extraordinary cases, symmetry breaking effects can be places where new physics are discovered. Now, in this biological context, what is exciting is that this electron bifurcation effect appears in the respiratory and photosynthetic cycles of, in fact, in the kingdom of life, we see it practically everywhere. This is one of the core energy conservation or transduction mechanisms that can be considered perhaps even on par with the use of ion gradients and ATP synthesis to use and convert and ultimately move energy around in physiology. Now, the signature property of electron bifurcation is that it enables reversible and nearly lossless coupling of the downhill and uphill electron transfer pathways. And furthermore, it suppresses short circuits, preventing the electrons from accidentally flowing backwards or, basically going the wrong way in the delicate operation of using the energetically downhill traveling electron to drive the other electron uphill in order to produce a more powerful oxidative state, ultimately for use in energy transduction further down the line. And it does this without sacrificing thermodynamic reversibility in a substantial way. And this is very interesting because in terms of classical physics, practically nothing occurs reversibly. Things tend to be, particularly in terms of energy transduction, very inefficient. So if we look at the history and the theory of bifurcation, Peter Mitchell's Q cycle came with the discovery of electron bifurcation during oxidative phosphorylation as an essential energy coupling mechanism. That was in the 1970s. Now, by 2008, Buckle and Thaler had discovered flavin-based bifurcation. Flavin had not been considered a bifurcating cofactor previously. They discovered this in anaerobes, and this revealed the fundamental physiological significance and biochemical diversity of bifurcating mechanisms. It was no longer just some kind of anomalous phenomena that we saw during respiration, that in fact, aerobic respiration, that in fact anaerobic life forms, simpler life forms, also used a different kind of bifurcation, showing that it's really an important process again across the kingdom of life. Now, by 2018, a structural comparison showed that electron bifurcating systems employ efficient long-range biological electron transfer to prevent back transfer, showing, at least at the most rudimentary level, the importance of quantum effects in these processes. And most recently, in 2026, an elegant mechanism revealed by photochemical experiments has shown increasingly the complexity and the sensitivity of the mechanistic control of this fundamentally important energy transduction mechanism. Now, I showed, or actually discussed, the two-flavin nature of the charge transfer state. These flavin-based bifurcating systems contain two FADs. And spectroscopic work has shown what initially was very interesting, a transient 726 nanometer band in the spectroscopic experiment was revealed during partial reduction. And this initially to the experiment suggested a delocalized charge transfer species spanning the two flavins, the alpha and beta FADs. So in this enzyme, FixAB, the extinction spectra showed that the 726 nanometer band appeared only at a specific combination of flavin redox states, as the electron transfer FAD is reduced and the bifurcating FAD is oxidized. And so the authors of this study initially proposed that as a form of long-range delocalization, but it would be particularly long-range in this case. And with more recent work, it has been somewhat conclusively established that the signature is now attributed to transient flavin-flavin contract. Once again, this is like a conformational gating effect where the two flavins move around, and when they get close enough, they can form this delocalized, highly characteristic 726 nanometer photochemical state. Now, what is interesting about this is that these classical conformational dynamics that gate the formation of the delocalized electronic state could occur with the characteristics timescale. And in fact, we expect it to occur on some characteristic timescale that is set by the protein's free energy landscape. And so the question is now whether this fluctuation just occurs randomly, basically turning off and on the delocalized electronic coherence, or whether there is actually a candidate possibility for what we call coherent control in quantum optics, whether there is actually some kind of knock-on effect from the characteristic frequency of the classical conformational dynamics and the coherent process involved in the delocalization effect. But as I have suggested, this is a highly debated topic, and certainly one that bears further experimental investigation. Now, if we look at this example of a bifurcating electron transfer flavoprotein, it has a pair of FADs, as I've described, and in another study, spin-dependent hydride transfer in bifurcating ETF was explored, and hydride transfer from cofactor NADH to the beta FAD was slowed by 1,500 times in the presence of a distant alpha FAD radical. This was tested against a radical FAD and simply a reduced FAD. And it was found that the spin nature of the radical, not its charged nature, appeared to control the rate over remarkably long distance for a radical mechanism. Now, once again, whether this is a true long distance effect or a conformational effect is not entirely clear. But what is interesting and fundamental from a quantum mechanical point of view is that normal kinetic isotope effect was found with the alpha FAD in comparison with an inverted kinetic isotope effect with the FAD radical. And this points toward the presence of some kind of tunneling mediated spin influenced chemical reaction, chemical mechanism in this bifurcating ETF system. Now, if we look at an even more sophisticated bifurcating enzyme, for example, the electron bifurcating nickel iron hydrogenase, there is a very extensive network of delocalized electron in the iron sulfur clusters of this system. In each iron sulfur cluster, in its reduced state at least, an extra electron is shared between two iron atoms in a single quantum mechanical state spanning both. So once again, we have this fundamental delocalization. That, as far as we can tell, the protein exploits in order to carry out the delicate operation of electron bifurcation. The bifurcating nickel iron hydrogenase, HYDE ABCSL, relays electrons through the iron sulfur clusters between the bifurcating flavin and catalytic nickel iron site. And so it actually is a very complex network, starting with NAD, nicotine amide, adenine dinucleotide, and flavin mononucleotide that transfers this electron through the iron-sulfur network and finally to the nickel-iron cofactor. And throughout this whole process, of course, the protein determines each cofactor's electronic properties, its coordination sphere and geometry, and the electrostatics, which control ultimately its redox properties, the reduction potential, and its redox role in the bifurcation mechanism. And of course, we expect that the system is not completely static in the sense it can only do one thing, that it can respond to environmental changes in order to adapt accordingly in order for metabolism to be regulated. So the study of these things have brought me, in my perspective, to a picture of what I would call biomolecular quantum control. To give the definition, quantum control is concerned with the active manipulation of physical and chemical processes on the atomic and molecular scale. And indeed, biochemical systems clearly can actively manipulate physical and chemical processes on that scale. So really now the question is, can we think of the protein as a quantum controller? In quantum optics, coherent control defines the use of a classical state to control a quantum state. For example, using a classical field of light, a coherent light field, in order to control some quantum process. And in biology, the protein environment modulates delocalized electronic states at the enzyme active site to achieve essential physiological outcomes: redox control, signaling, catalysis. And in this view, enzymatic proteins function as molecular scale quantum state controllers. The question is whether they enable coherent control in the sense that they have phase coherent control, that actually the timing or the synchronization of the classical protein state actually influences the quantum dynamics in a coherent way. And now, if we can answer that question, the next step would be to link it to homeostasis, to understand the role of molecular scale quantum control in the metabolic regulation of biochemical processes. And so in order to do this, we need modeling of these enzyme active sites. And of course, modeling enzyme active sites requires the treatment of electron correlation, delocalization, proton-electron coupling, and various electromagnetic interactions. So the question is not whether these quantum effects are there, but the degree of coherence that is innate to physiological regulation. Now, this has important implications for enzyme engineering and medicine. Without speculating any more about what we'll find, if we can harness the biochemical processes and, in fact, engineer them, for example, building artificial bifurcating enzymes, tuning the delocalization and spin environments for enhanced efficiency and reversibility, as these enzymes already do, we could drastically expand the catalytic toolkit that living systems already make available. This has, beyond, I think, tremendous industrial and agricultural applications. It presents many therapeutic and medical applications. For example, we might develop a DNA protective or redox modulating treatments to help address various metabolic diseases, pernicious diseases like cancer and other, forms of chronic illness. That we could begin to approach this by having an advanced picture of the cofactors and the ET pathways, electron transport pathways in these systems, and with that understanding, develop advanced design strategies, if I've made that clear. Finally, my own interest is in the quantum thermodynamics of these systems as well. Can we create quantum biological platforms to test and harness correlated reversible energy conversion in these systems, ultimately linking the fundamental quantum physics with practical applications, because of course these systems don't evolve at ultra low temperatures. They occur at physiologically ambient temperatures. And the question of the interplay between the quantum mechanics and the thermodynamics becomes very interesting in that regard. So what are some of the open questions in future directions? Well, I mentioned flavin oxygen chemistry is a major open problem. Flavin oxygen chemistry is very important to physiology, but not well understood. And so we want to know what structural and electronic conditions govern oxygen reactivity across the diverse landscape of flavor proteins. Can we predict and harness this for protein engineering? With, again, potent medical and therapeutic potential for applications. We have the question of how even to construct predictive design rules, which we currently lack. Can we derive general structural and, if not geometric structural, electronic structural principles for cofactor reactivity and, for example, a bifurcation or energy transduction design that enable rational engineering strategies for novel enzymes? And in this regard, we will need methodological benchmarks. These are community-wide benchmarks for modeling excited state, charge transfer, and spin-coupled mechanisms in simulations of protein enzyme environments. And the goal of these benchmarks will be to really define what constitutes a robust simulation of these systems, so that we don't change one parameter and find the predictive power goes away. We really want simulations that we know the full scope of its predictive power, that we can use consistently and rigorously over a wide range of related or even unrelated systems. And finally, the question of how to integrate this into our existing knowledge of systems biology. How do cellular conditions like pH, presence of metabolites, and other redox pools actually modulate physiological quantum control at active sites in living systems? What are the enzyme and system level impacts of each of the various physiological conditions? And I think these questions really are still just a tip of the iceberg, scratching the surface of the kind of deep questions that we can begin to ask about the inner workings of life itself. So, in conclusion, not just of Chapter 7, but of Part 1 of Physical Principles of Quantum Biology, I have developed, I believe, some unifying themes. And if you take away anything from your study of this part of the book, I want to convey that quantum mechanics operates throughout enzyme catalysis, from the polarized electronic structure that creates enzyme preorganization to the tunneling, vibronic coupling, spin chemistry, and photochemistry that contribute to these specific systems. These concepts are not separate biological categories, and they should not be considered ad hoc, so to speak. The polarized electronic structure that enables enzyme preorganization, the tunneling that crosses the residual barriers, and other ultrafast non-adiabatic electronic transitions in photochemistry are in fact just different facets of the same underlying quantum theory. And considering them all together, we can develop a unified theory of molecular biology and quantum biology in order to understand the deepest workings of anatomy and physiology. And so, in review, quantum mechanical principles discussed in Part 1 of the book provide a framework for interpreting and ultimately engineering various areas of enzyme catalysis, energy transduction, and in fact, various forms of photoreceptive and sensory properties of living systems as well. We've considered vibronic delocalization in photosynthesis, decoherent electron tunneling in respiratory chains, non-adiabatic photochemistry in rhodopsin, a photoreceptor of the eye, singlet-triplet interconversion in cryptochrome, the circadian photoreceptor that sets our daily biological rhythm, intersystem crossing in the DNA repair enzyme photolyase, and now in some depth, delocalized electronic structural effects for enzyme catalysis and energy transduction via electron bifurcation. But of course, this is really just the beginning of our study of quantum biology. We have now set the stage, if you will, to continue into a much more comprehensive study of the implications of these effects throughout biology. So if you've enjoyed our lecture today, please download your guide to quantum biology, Physical Principles of Quantum Biology, from the archive. It's available now for researchers, students, and curious minds ready to venture beyond classical biological explanations. Thanks again for your attention, and have a wonderful day. I'm Dr. B, and if you have any questions, please don't hesitate to send them to my email, babcock@gmail.com. And of course, you can check out the rest of the videos on my YouTube channel at Dr. Babcock. Thanks very much.